Yeah, thank you very much, Andrea. And welcome to today's webinar. So I'll be talking about human pathogenic fungi, and we start with an outline. So first I'm going to give you an overview of what human pathogenic fungi are. So fungal infections of humans vary in aggressiveness and severity. For example, a number of dermatophytes can cause superficial infections of skin and mucosa, such as, for example, the ringworm and the toe fungus. And these infections are fairly easy to treat. However, they also can cause more severe, more aggressive infections. Such as the subcutaneous infections, as shown here with the chromoblastomycosis, and also invasive, life-threatening, and often deadly infections that are caused by dimorphic and opportunistic fungi. And today I will specifically focus on Candida species, which are opportunistic fungal infections, causing opportunistic fungal infections. And the genus Candida comprises of about 150 species, but only a handful of those can colonize diverse host niches and cause infections in humans. And these pathogens are usually natural commensals of various host niches, such as in the GI tract, the oral cavity, and the skin of healthy individuals. But in individuals with weakened or compromised immune system, they can cause infections ranging from infections of the skin and mucosal surfaces to systemic and often deadly infections. So these infections are pretty difficult to diagnose and to treat due to the misdiagnosis or delayed diagnosis, and they can be caused also by multi-drug resistant species. And for these reasons, severe Candida species infections are on the rise. They are consistently ranked among the top 5 or top 10 nosocomial bloodstream infections in developed countries. For example, here report from USA ranks Candida species as the 4th most common nosocomial infection in the United States. And of these, Candida albicans is the most common cause of systemic candidiasis, although other pathogenic Candida species, for example, Candida glabrata, or the emerging global healthcare threat species Candida auris are also on the rise. So Candida albicans currently represents the model organisms to study the biology of other Candida species related infections. And as it has been the subject of extensive research for several decades for now, and it has been, it is the species that has the best developed genetic tools. And what we know about Candida albicans is that it lacks environmental reservoir, although sometimes it's found from plant-based substrates, such as here this report on oak trees. But normally it's very tightly associated with warm-blooded mammals and especially the female host. And it can, where it can inhabit a variety of host niches that present this fungus with diverse environmental conditions, including different nutrient conditions. And Candida albicans possesses an extended and sometimes unique arsenal of different proteases, transporters, enzymes, and molecular networks to allow for use of this wide array of host available nutrients. And some of these nutrient features can be exploited for antifungal drug development, and therefore they're very much worth investigating. So many pathogenic microorganisms possess such metabolic flexibility that is important not only for survival and growth, but also for stress and antimicrobial drug resistance, for immune avoidance, and for the expression of virulence characteristics. And Candida albicans is also one of those. Candida albicans, for example, can utilize or sense host nutrients to modify virulence, its virulence, for example, to switch to the more aggressive filamentous form, or to form biofilms and adhere to surfaces. So here I'm going to give you one example of how growth on host-relevant nutrients is connected to the persistence of Candida albicans in the host. So, for example, this is a classical example where cells grown on lactate, which is an abundant carbon source in certain host niches, such as in the vagina, have visibly different cell wall characteristics compared to the cells that are grown on glucose. And this makes the lactate-grown cells less visible to the innate immune cells, for example, macrophages. And therefore, the nutrient composition of the environment is directly connected to the capacity of this fungus to cause infection. And my group is very interested in understanding the links between nutrient utilization, stress resistance, and virulence in the human pathogenic fungi, with specific focus on Candida albicans, but now we are expanding also to other pathogenic Candida species. And now we know that metabolic signals are interconnected with virulence networks in these species. And I will proceed with one specific example of such link that we established with the help of the phenotypic microarrays. So now we go to the phenotypic microarray applications, and I will show you how we used them to screen for protein kinase mutant strains and find novel phenotypes. So Candida albicans, so in all micro and higher organisms, protein kinases coordinate responses to external and internal stimuli. And Candida albicans genome comprises of 108 protein kinases. And although some of these kinases have been characterized and functions have been assigned in various aspects of either Candida fitness or pathogenicity, the precise role of 59 of these kinases remains uncharacterized. And in order to comprehensively characterize the function of protein kinases in Candida albicans, our collaborators from University of Würzburg here in Germany have generated a complete kinase mutant library. So for this, we have duplicates, duplicate for each kinase. We have 86 homozygous mutant strains and 25 heterozygous mutant strains of possibly essential protein kinases. And so we proceeded with manual characterization, phenotypic characterization of these mutants, especially the ones that have no known functions or characteristics in Candida albicans. And we tested those for ability to resist stresses, various stresses such as oxidative or cell wall stress, to grow, to undergo hypomorphogenesis, or the susceptibility to antifungal drugs. But for a good handful of those, so nearly half of those 59 uncharacterized kinases, we still didn't have any known phenotypes. And here is a list of the kinases that we subjected to a phenotypic microarray analysis in order to screen for possible phenotypes and assign them some functions. And today I will present to you a pair of protein kinases. These are SCY1 and SCY2. Both are orthologs of the Saccharomyces cerevisiae. So the model is protein kinase SCY1. So in order to screen these kinases for kinase mutants for phenotypes, we proceeded with the standard protocol that was provided by Biolog. So we had stripped out strains, and we resuspended that in the media containing the redox dye in the Biolog plate. Pipetted those in the Biolog plates. And incubated in the Omnilog device, which holds 50 plates simultaneously, so it's able to measure in automatic manner the growth of these strains. So in each plate, we contained one strain and tested for multiple phenotypes. And here is the classical example of how the data is presented by the Omnilog device or using the available software. So here we can immediately see the growth curves of, for example, the wild type and the homozygous deletion strain of kinase of interest. And right away we can compare the growth characteristics of both strains. And you can appreciate immediately that the wild type strain grows quite better on most of the tested metabolites. So each well presents one metabolite, and we see that the deletion mutant has multiple growth defects. But this is one way to crudely look at the data. What we did is actually to use the available softwares, or we used available pipeline that is used for the statistical analysis of phenotypic microarray data, as was published recently. And here in brief, we have growth signals for each phenotypic microarray assay, and those were subtracted first from the respective negative control signals. And the negative values were replaced, if any, were replaced by 0. For each growth signal, we categorized it as active. And this was where the data fitted to logistic curve represented, which is representative to active growth, meaning active growth. And non-active in the case where a line without exponential growth phase characteristics was interpreted as non-active growth. And then in this case, potential growth is identified if not all replicates that were used for the data analysis were showing the same result. So, for example, we have one plate where we see active growth for the given metabolite and non-active growth for the other. And subsequent statistical analysis was used to further identify the significant growth differences per substrate between 2 or more strains. And with that, we were able to assign characteristics to multiple protein kinase mutant strains that were analyzed. And as I said already, I will focus today on one example. So these are the SCY1 and SCY2 predicted protein kinases in Candida albicans, and these are paralogous genes, SCY1 and SCY2. Both kinases or predicted kinases are uncharacterized in this pathogen. However, in Saccharomyces cerevisiae, which these kinases are orthologous to, SCY1 is the sole SR-like protein kinase in the species, where it's involved in processes such as mRNA metabolism and homeostasis. Of note, SR protein kinases in humans control essential cellular processes such as alternative and constitutive splicing. So for what we found with the microarrays data was that the SKY2 failed to grow on various TCA cycle intermediates, such as malic acid and succinic acid, for example. You can see the non-active growth for the mutant strain versus the active growth of the wild type. And then we confirmed the biolog generated data manually by growing the cells on various TCA cycle intermediates and comparing those with the upstream metabolic reactions. So for example, SKY1, and in comparison we used SKY2 mutant strains, could grow normally on pyruvate and glucose as carbon sources. However, as soon as we enter into the TCA cycle, we noted that the SKY2 mutants, so we used duplicate mutants generated independently. Both of those mutants failed to grow on alpha-ketobutyrate compared to normal growth for the wild type and SKY1 mutant. And for example, on succinate, we see the same effect. The same was seen also on malate and citrate, and the ones metabolite intermediates of the TCA cycle that are crossed out are the ones that do not support growth in Candida albicans, likely due to lack of transport in the cell. So clearly we see here that SKY2 mutant fails to grow on TCA cycle intermediates, and this is different from the SKY1, which has no defect in these conditions. This is another way that we visually present the biolog data, and here you can see also that SKY2 mutant has growth defects on various dipeptides compared to the wild type and the SKY1 mutant strain. So here you can see with purple the active growth of the wild type, for example. In this case, both the SKY1 and the SKY2 mutant fail to grow on this specific dipeptide. In this case, all three strains grow equally well or have active growth, although here we can also see whether this certain strain, for example, here the SKY1 mutant has growth and advantage over the SKY2 mutant, and the opposite here, the SKY2 mutant has growth disadvantage compared to the wild type. So even though in this case, always the growth is non-active. So with this color coding, we can easily see the differences in growth and the growth characteristics of certain strain and compare it to other. And in this case, potential growth represents the data set where one result was positive and the other one was negative. And a triplicate is required to really define whether this is positive or negative growth. So we verified also the these phenotypes for SKY2 as being defective in growth on various dipeptides, and we selected several dipeptide combinations, and we also used a tripeptide to compare the data. And we essentially were able to validate the phenotypic microarray data that SKY2 mutant fails to grow on various dipeptide combinations compared to the wild type or the SKY1 mutant, but no effect was seen on tripeptides. So for these reasons, we were able to conclude that SKY2 coordinates or mediates processes that are related to dipeptide utilization in Candida albicans, and these defects or the SCY2 defects are very specific and limited to dipeptides, as they are not observed with tripeptide combinations. We also did a phosphoproteomic analysis in order to identify SCY1 and SCY2 target or substrates, targeted substrates. And here are represented the number of proteins that are not phosphorylated in the mutant strains, but they're phosphorylated in the wild type. And so there is, you can see that SCY2 phosphorylates far more targets compared to SCY1, and that there is essentially very little overlap between the function of these two protein kinases. Of these 100 targets of SCY2, we see PTR22, which is a D and tripeptide transporter in Candida albicans, so very specific for transport of D and tripeptides. And we tested the PTR22 mutant strain as well as PTR22 overexpression strain in the wild type and in the SCY2 background. And what we see is that overexpression of this dipeptide transporter is able to rescue the phenotypic growth defect of SCY2 on these nutrients. So this validates that SCY2 really coordinates dipeptide transport in Candida albicans. We also tested the susceptibility of SCY2 mutant to dipeptide antimicrobials such as nicomycin Z and polyoxin D. And you can see that the SCY2 mutant shows severe defect on nicomycin Z, but not and even a higher defect on polyoxin D. So it's not susceptible to these antibiotics compared to the wild type, for example. So with that, I can summarize this part that we identified Candida albicans SCY1 and SCY2 to encode for SR-like protein kinases that are orthologs of Saccharomyces cerevisiae SCY1, and that their different SCY1 and SCY2 have different functions in Candida albicans, with SCY2 being involved in regulating metabolism of TCA cycle intermediates and in transport of dipeptides. Moving on to the next part, I will now show you or present you briefly how we used this phenotypic microarrays to identify species-specific differences in human pathogenic Candida strains. I noted before the top 5 pathogenic Candida species. So we used all those species, and as well as the very closely related and less pathogenic to Candida albicans species, Candida albicans. And in order to directly compare their metabolic characteristics using the biolog microarrays. Most of this was basically validation of previous observations by various groups that, for example, Candida albicans is very fit in utilizing a variety of alternative and primary carbon sources. You can appreciate the difference in variety and robustness of utilization of various carbon sources by Candida albicans compared to the other Candida species. But when we looked at the utilization of nitrogen sources, what we noted is that not only Candida albicans, but also Candida krusei and tropicalis are exceptionally tropicalis is very well fit in utilizing a whole variety of really a great variety of different nitrogen sources. In contrast, Candida glabrata was not as fit, and when we looked at the data, there is a slight hiccup here. So when we looked at the data, what we realized is that the data from the carbon sources is also not fitting very well. This is because here the nitrogen source is glutamine, and Candida glabrata, as you can see from here now, the nitrogen source plate does not utilize glutamine very well. And so for these reasons, we need to adjust the nitrogen source that is applied to the carbon source plates. For example, ammonium sulfate will be excellent candidate that should provide universal growth for all the different Candida species. And so when you're comparing different strains, it is very important to match the growth rates and also the base components of the medium need to be equal so that you're always normalizing to the equal amount of growth. So next, I'm going to show you how we used these phenotypic microarrays to look for clade-specific differences in human pathogenic Candida strains. I already introduced Candida auris. Candida auris is an emerging global healthcare threat that was first identified in 2009 in Japan. And because it's often multi-drug resistant, and in some strains or types, it's resistant to all three classes of antifungals, such as azoles, echinocandins, and purines. It's dangerous, and it has caused outbreaks in several major healthcare facilities, for example, in London and New York City. And here is a report from the CDC showing that the reported clinical cases of Candida auris in the past year with the start basically of the pandemic. And you can appreciate that the number of cases basically corresponds to the population distribution in the States. This is alarming because not only that this pathogen is multi-drug resistant, but it also can persist on patients' skin, clothing, and on surfaces. And it's commonly resistant or not really susceptible to common healthcare disinfectants. So it's really difficult to eradicate from surfaces, and sometimes it has caused a closing of whole hospital units for weeks or even months. So this causes also indirect costs to the healthcare, of course, and it's interesting to study this pathogen. What we know also about it is that there are 4 clades of Candida auris that originated almost simultaneously from different parts of the globe, and that we now know that the South Asian clade is responsible for the most severe outbreaks that were reported to date. So this is represented here. What we actually don't know is what is about Candida auris metabolism. So everybody is studying in detail the antifungal resistant mechanisms, but very scarce data on Candida auris metabolism is available. And I think it's important to link the metabolic properties to the antifungal resistance mechanisms. So, for example, multiple studies, as I mentioned, are studying the antifungal resistance. And as I mentioned already, the South Asian clade can rapidly acquire multi-drug resistance in vitro, as was shown very recently. Also, there is very limited data on Candida auris metabolism, especially regarding clade-specific metabolic characteristics. And for example, one study by Lockhart et al. Showed that isolates from India and South Africa, for example, are capable of assimilating N-acetylglucosamine or GLP-1, while isolates from Japan and South Korea were not able to process this compound as readily. And so when we ran so we took representatives from all 4 clades of Candida auris, so from Africa, East Asia, South American, and South Asian clades, and we subjected them to phenotypic screen using the phenotypic microarrays to look for fingerprints, metabolic fingerprints, or differences that can assign some characteristics to these clades. And so, for example, we were able to quickly verify that indeed African and East Asian clade are incapable or less fit to grow on N-acetylglucosamine, whereas the South American and South Asian strain clade isolates are well fit to do so. So this was, again, very reassuring. And so when we started looking at specific differences between the clade isolates for growth on various compounds, we noted that, for example, Candida auris different isolates utilize a very small range of different carbon sources, so very limited repertoire. But some of those show some specific differences. And overall, what we also saw is that, for example, the African clade isolate displayed overall better growth on carbon sources. So always the curves were slightly higher compared to all the other clades. So this was an interesting observation, but another interesting observation is the variety of different peptide nitrogen sources. And here is just one example, but this was seen across the board with all nitrogen sources essentially, that, for example, the South Asian clade is exceptionally well fit to utilize a variety of different dipeptides and as well as more complex and nitrogen sources as well as amino acids. So perhaps this ability to utilize a variety of nitrogen sources is connected to somehow to the highest capacity to acquire multi-drug resistance and to cause infections in susceptible individuals. So this study is still undergoing. What you see with the black is unknown status, where, of course, one set of data showed active growth, the other non-active growth. And now we are running a triplicate to define and fill in the gaps, essentially, and have the comprehensive set available to the public. So in summary, what I showed you here is that phenotypic profiling allows us for rapid visualization of clade and species-specific metabolic features, that utilization of extended number of nitrogen sources might provide an edge in pathogenicity, and that the susceptibility to antifungal peptides, for example, or alternative small antibiotics could be tested to really verify these findings. Since these antifungal peptides will represent similar nitrogen sources that are readily utilized by certain clades or species and not so well by others. And now I'm moving to the third and last application of phenotypic microarrays that I would like to present to you today. And this was done in collaboration with excellent bioinformatician from the group of Janik and Diotu. And this is collecting we collected phenotypic data to aid the precise modeling of metabolic exchange between Candida albicans and the members of gut microbiota. So here, what was done by our collaborators is actually to generate a genome scale Candida albicans metabolic model, where they started with a model that was automatically generated by comparative reconstruction pipeline or Core pipeline. And in this initial model, it was comprised of 3,298 reactions, of which 3,150 were network and 148 were transport reactions. And this also represented 2,770 metabolites. And next, what they did is to resolve the invisible energy generating cycles, which created, for example, energy compounds such as ATP without requiring nutrient uptake. And by either changing the metabolite usage or reaction directionality that was based on the TEC or biosys information, they were able to correct for several reactions. So then we came into play, and they asked for available by or provided by us biolog data, and also used published biolog data on Candida albicans, where they took these data sets and also and were able to refine these predicted interactions, essentially. So we used data from wild type strains, as well as we generated additional data sets from metabolic knockouts, for example, for arginine biosynthesis. And this data was used to refine the model. And essentially, this also helped to remove some faulty interactions or generate new connections. And at the end, this led to a refined model. And here you see the outcome is different from the starting outcome, in which we have corrections for we have now 3,082 reactions and 234 transport reactions, compared to a much larger number compared to before. So this was this model was then used by them. So with this model now they were able to achieve over 90% accuracy between the prediction model and the phenotypic microarray growth experiments, and this was done precisely for carbon, nitrogen, and sulfur sources. Model was limited to those compounds. And next, they generated this in silico metabolic interaction predictions about Candida albicans that is coupled to gut bacterial genome scale metabolic models that were available for 910 genome scale metabolic models in bacteria. And this was achieved by pairwise metabolic analysis by linking the Candida albicans model to each gut microbe model. So with this, they simulated growth on two different media compositions. So for example, the Western and high fiber diet. And this helped us, to show that majority of the gut bacteria have either basically mutualistic or parasitic relationship with Candida albicans, but also some different interactions were seen, such as commensalism in this case. And among all these studied microbes, they stumbled on one interesting example, and this was Alistripes putresciens, what was predicted and then confirmed via in vitro and also in vivo data that negatively affects Candida albicans levels in the gut. So you can appreciate how going from predictions and some with the help of some experimental data, they were really able to go and validate these specific interactions in the gut. So in summary, screening of Candida albicans protein kinase mutant strains was able to reveal novel phenotypes, such as the role of SCY2 in metabolic adaptation. There are some other examples that I could not show today. Isolates from different pathogenic Candida species, as well as various clades from the same species possess unique metabolic profiles that are frequently linked to strain fitness, essentially. And genomic scale metabolic models can be used to make precise predictions of metabolic interactions between members of microbiota or the host, only paired with experimental data. So experimental data is really required to refine and assign precise predictions to experimental outcomes. With this, I would like to thank first and foremost my group, and especially Philip Frank, who launched the run of all these phenotypic microarrays, and really this is a lot of this work is due to his contributions to this research, as well as other members in the lab that either directly or indirectly are working on the listed projects. Also, our collaborators from the Hans Kniol Institute in Vienna, Germany, and University of Wolfsburg, again in Germany. And thank you for your attention. I'll be happy to take your questions.